Right now, in a laboratory in Beijing, a liver is growing inside a petri dish. It is not a full liver. It is smaller than a pea, but it produces bile. It metabolizes drugs. It does exactly what a human liver does, outside of a human body, built from scratch, from cells. This is called an organoid, and China is producing more research in this field than any other country in the world. The question is no longer whether growing organs in a lab is possible. The question is who gets there first. And the answer emerging from the scientific data of 2024 and 2025 keeps pointing to the same place. The problem that never changes. Right now, more than 100,000 people are on the organ transplant waiting list in the United States alone. 13 of them die every single day without receiving one. In China, the ratio is even more brutal. 30 people waiting for everyone who receives. Kidney, liver, heart. Medicine figured out how to transplant them decades ago. It never figured out how to produce enough of them. Human biology does not change. People die faster than they donate. And the waiting list grows every year. For decades, the only answer was more donation campaigns, different consent laws, more efficient organ banks. All of that helped at the margins. The core problem remained. Then a group of scientists started asking a different question. What if we didn't need donors at all? What an organoid is, and why it changes everything. In 2009, a Dutch researcher named Hans Clevers managed for the first time to make human intestinal cells self-organize into a three-dimensional functional structure outside the body. That was the first organoid. The core idea is that human cells carry within them the instructions to build the organs they belong to. Inside a body, those instructions are activated by the surrounding environment. In the lab, scientists learned to recreate that environment, the right temperature, the right chemical signals, the right physical support. And the cells do the rest. They grow, they organize, they form structures that work. Since 2009, organoids have been created for the kidney, liver, lung, pancreas, stomach, brain, intestine, and heart. None of them are large enough for transplant yet, but all of them function. What China built China did not invent the organoid, but it is dominating what comes next. At the Chinese Academy of Sciences in Beijing, researchers developed liver organoids using chemically reprogrammed stem cells, called HCiPSCs, that do not require viruses to be created, making the process safer and far more scalable. These organoids were mixed with a biocompatible hydrogel and printed in layers using 3D bioprinting, a printer whose ink is living cells. The result was transplanted into mice with severe liver failure. The animals survived. The tissue integrated. It functioned. It did the work of the liver that had failed. This happened in 2024, published in Science Advances, one of the most rigorous scientific journals in the world. Not in humans, but the step directly before humans, Tsinghua, and the most sophisticated liver ever built. In 2025, researchers at Tsinghua University, China's MIT, published something that redefined what is possible in tissue engineering. They created a liver organoid that replicates not just the structure of the organ, but its internal metabolic zonation. That requires some explanation. A real liver is not uniform. It has different zones with different functions. One region that metabolizes alcohol one way, another that processes drugs differently, a third that regulates protein production. Till 2025, no organoid had managed to reproduce this. The Tsinghua organoid did. By using signals from specific molecular pathways, T and Notch, to control which genes are activated in each region of the tissue, the team created a miniature liver with genuine functional architecture. Not just liver cells growing together, a liver functioning like a liver. The problem that still needs solving, if all of this is possible, Why is there still no lab-grown organ being transplanted into humans? The answer has one name: vascularization. An organ is not just cells. It is cells connected by a network of blood vessels that delivers oxygen and nutrients to every point in the tissue. The heart has capillaries reaching within 0.1 millimeters of every cell. The liver has a three-layer interconnected vascular architecture. The kidney has microscopic filtering structures that no printer can yet replicate with enough precision. Printing the outer structure of an organ is possible. Printing the internal vessels with the density and precision needed for the organ to survive inside a human body, that is the unsolved problem. And that is exactly where China is focusing now. In Shenzhen, the Institute of Synthetic Biology at the Chinese Academy of Sciences is developing high-resolution bioprinting techniques capable of creating vascular networks inside printed tissues. In Shanghai, research groups are using microfluidic chips to simulate blood circulation inside organoids, keeping them alive longer and with more function than any previous method. Why China specifically? In April 2025, China did something no other country had done. The National Science and Technology Ethics Committee published the world's first comprehensive ethical guidelines for human organoid research. This included specific rules for brain organoids, synthetic embryo models, and chimeras, structures that combine human and animal cells. While the rest of the world is still debating how to regulate this technology, China created the framework, set the boundaries, and is already operating within them. That is not coincidence. That is strategy. According to the 2024 Nature Index, China ranks second in the world in health sciences research output, behind only the United States. In biotechnology and regenerative medicine specifically, the volume of Chinese publications has doubled in the last 5 years. And behind those numbers is a pressure that Americans and Europeans do not feel in the same way. 1.4 billion people, 30 on the waiting list for everyone who receives an organ. A public health crisis that makes the urgency of solving this problem unlike anywhere else in the world. What comes next? Scientists working in this field speak in phases. The current phase, which China dominates, is functional tissue in the lab, organoids that work, tissues that integrate in animals, structures growing larger with every generation. The next phase is vascularization solved. When that problem is cracked, the path to human transplant opens. The phase after that is scale, not just one kidney grown for one specific patient, but the possibility of producing organs on demand from the patient's own cells with no rejection risk and no waiting list. Professor Shubo from the Shenzhen Institute of Synthetic Biology described the trajectory directly. Bioprinting functional organs for human transplant is not a question of if, it is a question of when. And the window he and other researchers cite is somewhere between 10 and 20 years. In a laboratory in Beijing, a liver smaller than a pea produces bile, metabolizes drugs, and does the work of a human organ. It is not ready to save a life yet, but it is working, and every generation of that experiment is larger, more complex, and closer. The organ transplant waiting list killed people yesterday. It will kill people tomorrow. The question those Chinese laboratories are trying to answer is, for how much longer? If this video showed you something you genuinely did not know, a like and a subscription helps this channel reach more people. And here is the question I want to leave you with. What do you think will be the first complete lab-grown organ transplanted into a human being? A liver, a kidney, or a heart? Drop your answer below. I read every single one.